The hypoxic insert device has 6 pillars that nest into the wells of a 6-well plate. Gas flows across the microfluidic network at the base of the pillar and can diffuse across the gas-permeable PDMS membrane separating the gas network and culture media. This device minimizes the diffusion path length between the oxygen source, the microfluidic network at the base of the pillar, and the cells, permitting more rapid oxygen concentration equilibration. The device also permits far greater control over the spatial pattern of oxygen exposed to cells, which is determined by the microfluidic gas network design made by standard photolithography using photomasks.
Hi, I'm Shawn Oppegard from the Dr. David Eddington lab in the Department of Bioengineering at the University of Illinois at Chicago.
Hi, I'm Elly Sinkala, also from the Eddington lab.
Today we will show you how to fabricate, validate, and utilize a custom device to expose in vitro cell cultures in a 6-well plate to different oxygen concentrations.
We use this procedure in our laboratory to study the effect of oxygen on wound healing, reactive oxygen species generation, and migration. So let's get started.
To begin fabrication of the oxygenating insert, prepare polydimethylsiloxane, or PDMS, in a 10:1 ratio between prepolymer and curing agent, according to manufacturer's instructions. The PDMS mixture is added to a previously machined Delran mold, patterned to mimic the shape of a standard 6-well culture plate. Once cured at 75 degrees Celsius overnight, the resultant replica molded pillar array will form the first of 3 components that comprise the oxygenating insert. In order to